All right, so today we are going to look at the structure of hemoglobin as well as its functions. Our lectures on hemoglobin, I've broken it into two. Today we are just looking at the structure and the functions and some hemoglobinopathies, that is some abnormalities in the structure of hemoglobin that would result into some health conditions. In the second part, which hopefully we'll be doing next week, we'll be looking at some more hemoglobinopathies that are not related to the structure of, say, the protein part of hemoglobin, but rather the heme part of hemoglobin. So today we start with the structure of hemoglobin. We know hemoglobin to be the pigment in blood that is responsible for the red color of blood. And that is to say that hemoglobin is a red pigment. Of course, this we find in erythrocytes, that is in red blood cells. And we also know the function of hemoglobin to be the carriage of oxygen from the lungs to the tissues, and then carbon dioxide from the tissues to the lungs. So we delve in deeper to see how this structure looks like. So hemoglobin is referred to as a tetramer of proteins, or is a tetrameric protein. And what that means is that we have the structure of hemoglobin having 4 proteins that are bound together, acting as one unit. So we see the alpha and the beta proteins bound together with the heme, which is represented in these red boxes. So the heme is bound to each of these protein structures. Now you might be wondering, what is this about alpha 1, alpha 2, beta 1, beta 2? If you remember your biochemistry from first year, you had looked at the secondary structure of proteins. And some of these structures you saw were alpha helix and beta sheets. So this is what the alpha and the beta chains of the hemoglobin is about, meaning that in this structure of hemoglobin, you are having 2 alpha helixes and 2 beta helixes, and you have 1 alpha bound to 1 beta, and the alpha 2 also bounded to beta 2. And each of these alpha and the betas are also bound to each other, with the heme being the prosthetic part. Again, you studied enzymes and you learned that the protein part of enzymes is what is referred to as the apoprotein, and the prosthetic part is the non-protein part, which can be some organic compound which is acting as a coenzyme, or some inorganic metal ion which is acting as a cofactor. And these parts which are non-protein are referred to as prosthetic parts. In the same way, in hemoglobin, we have the prosthetic part, or the non-protein part, being the heme. And the globin is the protein part, which are the alpha and beta globins of hemoglobin. Now, the most common form of hemoglobin you will find, we will soon see others, is the hemoglobin A1, which is made up of 2 alpha chains and 2 beta chains. That is actually the structure that we are looking at, the alpha and beta chains. Now, one alpha chain contains 141 amino acids, whilst one beta chain contains 146. And so in total, this will be giving you 574 amino acids in the structure of hemoglobin. I think in this equation here, we should have gotten 2 times alpha and then 2 times beta. As you see in the structure, there are 2 alphas and 2 betas. So that if you do 2 times 141 and then 2 times 146, and you add together, you will have 574 amino acids, being the number of amino acids which are found in the structure of hemoglobin. Now, how about the heme? How does a heme look like? So the heme is also, let's say it is referred to as a complex of 4 chiral rings. So if you look carefully at each of these rings, A, B, C, and D, you find out that the rings are similar, having a nitrogen atom at one of the points of the ring, the 5-sided ring. Now, each of these rings are bound together with some methanol bridge. So in between A and B, you find some methanol bridge. In between B and C, you have some methanol bridge. C and D, the same. And then you have between D and A. So these are 4 rings bound together with a bivalent ion at the center, which is binding these nitrogens in these spiral rings together. So we have this representation in green as the protein part. So this is, so histidine globin, this is just showing that this is an amino acid. And then, so this will be a side chain, and you have the carboxyl group at the other end. So we have the protein, which is bound to this ion at the center of the heme. Of course, we have the letters which are in red, being various side chains which are bound to these rings. So the M refers to methyl groups, and then the V refers to vinyl groups, whilst the P refers to propionyl groups. So these are the different groups that are found on the chiral rings, the 4 chiral rings that form the full structure of heme. So, like we were saying from the beginning, that we have the HB1 being the most common form of adult hemoglobin, where we have 2 alpha globins and 2 beta globins bound together. Of course, this is about 90% of all the hemoglobin in the body. We'll come to see that we also have hemoglobin A2, which is made up of 2 alpha globins and 2 delta globins, but these will form less than 5% of all the hemoglobin in the body. We also have the fetal hemoglobin. So this fetal hemoglobin is what is found in the fetus, and this is less than 2%. In fact, after birth, the synthesis of this fetal hemoglobin ceases, and the adult hemoglobin alpha and beta take over. Remember, respiration in the fetus is such that the fetus does not breathe in oxygen, but rather gets the oxygen through the placenta from the mother. And so we have this special hemoglobin alpha, gamma, less than 2% in adults, but in fetus, that is the major hemoglobin you find, which is carrying oxygen to the baby, and then carbon dioxide back through the placenta. Then we also have the glycosylated hemoglobin. Those of you working in some clinical settings, you would have come across this glycosylated hemoglobin test, which currently has replaced this fasting blood sugar, so that you don't necessarily have to go take this fasting blood sugar test in the morning without eating. But we have hemoglobin that is bound to some amount of glucose in the body, such that in the conditions of diabetes, you have these glycosylated glucose increasing in elevated amounts, so that if this glycosylated hemoglobin is determined in the blood and the levels are higher than normal, then that is a diagnosis for some diabetic condition. In general, the total of this glycosylated hemoglobin is less than 5% in the body. Now, apart from hemoglobin as this hemoprotein in the blood responsible for the cellular respiration, we also have myoglobin. Now, myoglobin you can refer to as a monomer of a hemoglobin. So what that means is that you have myoglobin being just a globin protein with heme attached to it, so that you have this myoglobin, especially in the heart and skeletal muscles, for the carriage and some temporal storage of oxygen in the blood, such that if you have hemoglobin being made up of 4 monomers, meaning that that hemoglobin will be able to bind to 4 atoms of oxygen, or 4 molecules of oxygen, 4 molecules of oxygen for hemoglobin. You have for myoglobin just 1 molecule. So it is only a monomer. It's a monomeric hemoprotein, and this is made up of 153 amino acids. So that's the difference. That the function of hemoglobin is for the transfer of oxygen from the lungs to the tissues and carbon dioxide from the tissues to the lungs. Hemoglobin does not store oxygen, but myoglobin has a temporal storage, and it also serves as an intermediary, as we'll soon see, an intermediary in the transfer of oxygen to the tissues when the hemoglobin carries the oxygen from the lungs to the tissues. In the tissues, myoglobin serves as a temporal intermediary for the transfer of the oxygen to the tissues. Now, before we continue further, let's look at the binding of oxygen to hemoglobin and myoglobin, the differences in these two. As we were saying earlier, that one hemoglobin will carry 4 molecules of oxygen, whilst one myoglobin molecule will carry 1 molecule of oxygen. So it means, yes, in myoglobin, you are just having 1 heme in that molecule. Now, what will be the difference in the saturation of these two blood pigments? So when you look at this graph, you see the saturation of oxygen on the vertical axis, and this is on the horizontal axis. You have the partial pressure of oxygen. You can say the concentration of oxygen. So what this means is that for myoglobin, you see the graph is very steep and has reached saturation earlier than for hemoglobin. And that is understandable because you just have one molecule of oxygen to make myoglobin saturated. It is just one monomer of a protein and a heme bound together for myoglobin. So once one molecule of oxygen binds to it, myoglobin becomes saturated. And so myoglobin gets saturated faster. So you see the graph being very steep. On the other hand, when you look at hemoglobin, see hemoglobin gets first bound to one molecule of oxygen, 2 molecules, 3 molecules. And so you see that the saturation process is gradual before it reaches saturation, where you have all the heme molecules being bound to 4 molecules of oxygen. And so you realize that this gets saturated at a much lesser rate than for myoglobin. And of course, the saturation is happening in the lungs. So saturation of oxygen happening in the lungs. And then you have here, down here will be in the tissues where oxygen is released from the hemoglobin. Okay, now, the term cooperative binding of oxygen to hemoglobin refers to the fact that the binding affinity of heme in hemoglobin to oxygen increases with the binding of oxygen to other hemes, so that if we take, this is the heme molecule, this is not bound to any molecule of oxygen. So this is deoxyhemoglobin. Now you realize that when one molecule of oxygen comes to bind to one of the heme molecules in hemoglobin, and that increases the affinity of hemoglobin to oxygen, and it goes in for the second, and the affinity increases the more to go in for the third, and also for the fourth, for hemoglobin to become fully saturated, become an oxyhemoglobin. Now, the reverse also happens, so that in the tissues, when the first oxygen is being released, first molecule of oxygen is being released to the tissues, then the affinity of hemoglobin to oxygen reduces. And so it releases a second molecule of oxygen, and it's left with 2. And the process continues until all the molecules of oxygen are released to the tissues. Now, that is the cooperative binding of oxygen to hemoglobin. Okay, I see Evans Appiah has raised his hand. Yes, Evans.
Hello, sir. Please.
Yes.
Please, can you go over the increasing affinity, please?
Increasing affinity. You mean on this particular slide?
Yes, please.
Okay. So we are looking at the cooperative binding of, okay, Shadrach, I've seen your hand. So when I finish, I'll call you. So we are looking at the cooperative binding of oxygen to hemoglobin. And we say the binding of oxygen to the heme in hemoglobin increases when you have oxygen bound, when you have the first molecule of oxygen coming to bind to the hemoglobin. So this is for the deoxyhemoglobin. There is no oxygen bound to it. You have a first molecule of oxygen coming to bind to the hemoglobin, and the affinity of hemoglobin to oxygen increases. So the affinity of this monooxyhemoglobin for oxygen is higher than the affinity of the deoxyhemoglobin. So it goes in for a second molecule of oxygen. Then the affinity increases the more. So all this process we are describing is happening in the lungs where there is saturated amount of oxygen. Then it goes in for the third. The affinity increases the more, and it goes in for the fourth molecule of oxygen. Now the hemoglobin is saturated. Then, of course, the reverse of this process is what happens in the tissues when oxygen is released from hemoglobin to the tissues, so that when the first molecule of oxygen is released to the tissues, affinity reduces the more, it releases the second, and then it releases the third, then it's left with the last, which it also releases to the tissues for hemoglobin to become deoxyhemoglobin. Evans, is that understood?
Well understood, sir.
All right. So Shadrach, I saw your hand.
Yes, sir. I was about saying the same thing, but Evans has already asked.
Okay. All right. So we can continue. So, okay, Alex.
Okay, so can we say the forward process is for saturation and the reverse process is for unsaturation?
Exactly. So the forward process is for saturation of hemoglobin and the reverse is for desaturation. Yes, you can say that. All right. So since we were talking about the affinity of hemoglobin for oxygen, the binding of hemoglobin to oxygen and its saturation, based on that, there are two forms of hemoglobin. And these are the deoxyhemoglobin. Deoxy meaning they are not bound to oxygen. And then we have the oxyhemoglobin. These are bound to oxygen. So when you look at this pictorial representation, we have the heme part of hemoglobin being this middle part, which is red. So this is an alpha globin. This is also an alpha globin. This is a beta globin. This is the second beta globin. Then we have the oxy. When you look at the oxyhemoglobin, we see the oxygen molecule being bound, which is here in blue. This blue bound to the heme of the hemoglobin. These are oxygen molecules. Now, these two forms of hemoglobin, that is the deoxy and the oxy form, are also referred to as the T form and the R form. So the T form referring to TOT or TENS, that is for deoxy, Now you see that the TOT or the TENS form, this is a form of hemoglobin that you see that the hemoglobin monomers are tightly bound together. These are tightly bound together, and you see there is a strong hydrophobic bond between the alpha and the beta globins, and as well as some weak ionic and hydrogen bonds between the alpha globins. They are tightly bound together. That's why they are referred to as TOT or TENS. No oxygen bound to them. Now, like we were saying, that when you have oxygen coming to bind to hemoglobin, it increases the affinity of hemoglobin for oxygen the more. And that is because some conformational changes occur in the hemoglobin molecule when oxygen is present, so that the hemoglobin molecule now becomes relaxed. So you see the bonds in between these alpha, these are the weak ionic bonds which you find here between the alpha globins get disrupted. So oxygen disrupts these bonding. Of course, you have these strong hydrophobic bonds between the alpha and beta dimers remaining, but you find that these ionic bonds, the weak ionic and hydrogen bonds between the alpha globins are disrupted, and so this hemoglobin molecule becomes relaxed. That is an oxyform, and that, of course, causes high affinity hemoglobin or oxygen. Any questions? I thought I was hearing somebody's microphone on. Okay, all right. So,
Sir, please go over.
okay, so what I was saying is that we have two forms of hemoglobin, the T form and the R form. So the T form is what is referred to as the taut or the tense form. The taut or tense form because the hemoglobin dimers, so if I say a dimer, I mean alpha, beta bound together by these strong hydrophobic bonds. That's a dimer. Now, so which means that we have alpha, beta dimer, two of them. Now, in between these two, there is some weak ionic and hydrogen bonds binding the alpha globins together. And these weak and ionic bonds get disrupted by oxygen when oxygen binds to the heme to form the relaxed form, which is also referred to as the R form, which is the oxyform. Oxy because the hemoglobin is now bound to oxygen molecules. And so the affinity increases because this hemoglobin molecule now becomes relaxed due to the conformational changes which occur when oxygen binds to the hemoglobin molecule. Is that clear?
Yes, sir.
All right. So now let's take a look.
Yes, sir.
Somebody talking?
Yes, sir. Yeah.
Right.
I would like to ask this question: whether, whether temperature has effects on oxygen binding to the.
Whether what?
Temperature has effects on the oxygen binding to the hemoglobin.
Okay, whether temperature has effect on the binding affinity of oxygen to hemoglobin. We'll come to look at the allosteric effectors of hemoglobin. So the factors which are responsible for the increase or decrease of affinity of oxygen to hemoglobin. Of course, temperature is not part of them, but what temperature may do, just as temperature will do to any protein structure, is that if you have high increase of temperature above normal body temperature, it will cause denaturing of proteins. That will happen in severe conditions of fever. But temperature directly does not affect that binding affinity of oxygen. Is that clear? It's not one,
Yes, sir.
temperature is not one of those allosteric factors of oxygen binding to hemoglobin. Is that clear? I saw somebody's hand up earlier. Okay.
Sir,
Yes,
sir,
yes.
please can you go to the next slide?
There's what?
Sir, please can you go to the next slide? I have a question there.
The previous slide, this one.
Oh, sorry, the previous one. not this. Yes, this one.
Mhm.
So my question is that the oxyhemoglobin,
Yes.
does it also have the strong hydrophobic bond between the alpha and the beta?
Yes, so I mentioned that the dimers remain together. As you see, the strong hydrophobic bonds remain. It is the weak ionic and hydrogen bonds between the dimers, that is specifically between the alpha globins, that are disrupted. Is that clear?
Yes, sir. Thank you.
All right. Any other? Okay. So we go on with the transport of oxygen and carbon dioxide, as we talked about earlier. So this is a general overview of what is happening. So this is in the lungs. Oxygen is breathed in, so there is high saturation of oxygen in the lungs. And these oxygen molecules get bound to hemoglobin to give you the oxyhemoglobin. So you see the 4 molecules of oxygen that are bound on hemoglobin. Hemoglobin then carries these oxygen molecules to the tissues. And you see here that myoglobin is serving as an intermediary here for the transfer of oxygen to the tissue. Charles and Ruja, you have anything to say, Charles? All right. So we see myoglobin here serving as an intermediary for the transfer of oxygen from hemoglobin to the tissues. Then, of course, the byproduct of cellular respiration, being carbon dioxide, gets bound to hemoglobin to form the carbamyl hemoglobin, and then it's released back in the lungs to be exhaled. Of course, we'll come to see that it is not all the carbon dioxide that get bound to hemoglobin directly. We'll soon see that to be exhaled in the lungs. So of course, as you know, yes, in the lungs, there's high concentration of oxygen. And so once one molecule, like we saw, with affinity of hemoglobin to oxygen increasing, one molecule binds to it, affinity increases, and so on and so forth. And then you have all the 4 molecules binding to the hemoglobin before it's carried to the tissues. So you see, as I was saying, it's just about 50% of carbon dioxide in Isaac entry.
I'm sorry, sir. Sorry, sir.
Okay, so I was saying that it's just about 15% of all the carbon dioxide that is released in the tissues that gets directly bound to hemoglobin to be taken to the lungs. I'm trying to minimize this and it's not going. Let me see. So let's please try as much as possible that we don't disrupt the lecture with our microphones on. As I try to mute everyone, now this window is refusing to leave. Okay, let's see. We can't even go to the next slide. I'll stop sharing briefly to deal with the issue and then come back. Okay. All right. So the rest of the 85% of carbon dioxide, what happens to it? Since we were saying from our previous slide that only 15% of the carbon dioxide gets bound directly onto hemoglobin to be carried to the lungs to be exhaled. Now, this is what happens to the rest of the 85%. The rest of the 85% of carbon dioxide gets bound to water. So there's a reaction between this carbon dioxide. There's a combination between carbon dioxide and water to give us carbonic acid. Now, of course, this is an enzymatic reaction being catalyzed by the enzyme carbonic anhydrase. Now, this carbonic acid further breaks down into a bicarbonate ion and some hydrogen ions. So this is happening in the tissues because, of course, the carbon dioxide is being released as a byproduct of cellular respiration in the tissues. Now, you see that we are talking about hydrogen ions, and that quickly should make you think about increase in pH in the cell. We'll come to that. Now you see in the lungs that carbon dioxide is being exhaled, and you see a reverse reaction. So this reaction where you have carbon dioxide and water binding to give you the carbonic acid. You see the reverse of this happening to give you back the carbon dioxide and water, which is exhaled as carbon dioxide and water vapor. Now, in the lungs, where you have 4 molecules of oxygen coming in to bind to hemoglobin. So 4 molecules of oxygen come, bind to hemoglobin to give you the oxyhemoglobin. Here, this oxyhemoglobin is then carried to the tissues. And whilst this oxygen is being released to the tissues, you have these hydrogen ions binding to hemoglobin so that the rest of carbon dioxide, which is not binding to hemoglobin to be carried directly to the lungs to be exhaled, rather binds to hemoglobin as protons or as hydrogen ions. So you have this bicarbonate ion moving freely in plasma, whilst you have these hydrogen ions or protons binding to hemoglobin to be carried to the lungs. So that back in the lungs, you have the hydrogen ions being released from hemoglobin, whilst it gets bound to the oxygen. And then you have these bicarbonate ions in plasma combining back with the hydrogen ions to give you back the carbonic acid, which is broken down further into carbon dioxide and water to be exhaled. Any questions?
No, please.
Okay.
Sir,
All right.
please, can you go back again?
All right. So what I was saying was that we saw earlier, so from the previous slide, this is the previous slide. The last statement here is that 15% of carbon dioxide in blood binds with hemoglobin. So here in the tissues where you have carbon dioxide. It's shown here that carbon dioxide is binding to hemoglobin to be carried to the lungs to be exhaled. And we say it is only 15% of this carbon dioxide binds directly like that with hemoglobin to be exhaled in the lungs, which means we are left with 85% of carbon dioxide produced in the tissues to be exhaled. Now, that 85% of carbon dioxide binds with water to form carbonic acid. And that is an enzymatic reaction catalyzed by the enzyme carbonic anhydrase. The carbonic acid further dissociates into a bicarbonate ion and hydrogen ions or protons. So if you say hydrogen ions, and sometimes in some books, they will refer to it as protons. That's why I'm saying hydrogen ions or protons. All right. So you have this happening in the tissues. Now, we have these hydrogen ions coming in to bind with the hemoglobin that has come into the tissues to release oxygen to the tissues. So hemoglobin, you have this oxyhemoglobin coming into the tissues to release oxygen to the tissues and then carry carbon dioxide away. So the 85% of that carbon dioxide, which it should carry away, is now binding to the hemoglobin to give you this hemoglobin proton complex. Now, this hemoglobin proton complex, or let's say protonated hemoglobin, this protonated hemoglobin now moves in the blood to the lungs. Now, these protons that are binding to the hemoglobin, okay, Kidir, I've seen your hand, so I'll call you. This protonated hemoglobin moves to the lungs, whilst we have the bicarbonate ions flowing freely in plasma. So now you are in the lungs, and here in the lungs, you find oxygen that is breathed in, coming in to bind to the hemoglobin to give you the oxyhemoglobin. And then the protons on the protonated hemoglobin are released in the lungs, where they bind back to the bicarbonate ions, which are flowing freely in plasma. And these two combine back. So this will combine back to give you, so this is a reverse reaction of what is happening in the tissues. So this will combine back to give you the carbonic acid, which further breaks down into carbon dioxide and water and exhaled as the carbon dioxide gas and water vapor in the lungs. So I was seeing a couple of hands when I was doing the explanation. So let's see those hands, and then, okay. So first, dear.
Say, please, sorry to interrupt. Some people are in the waiting room.
Some
They
people
are not
are,
able to join. Yeah, they said the meeting has been logged.
the meeting has been locked.
Yes, they said they are not able to join.
Actually, there is no waiting room enabled. So I don't really know what they are talking about. Meeting info. Because anybody at all could enter at any time.
I have a question.
Allow participants to place on. You see, there is no, okay, lock meeting. All right. Okay. So I think those who are locked can enter now. And who else? I think somebody else's hand was up. No?
I have a question.
Okay. So, okay, let's see.
So please,
Jose.
yeah.
Yeah, sir. I realized that the bicarbonate ions
Mhm.
that are in the tissue, I realized that they are left in the tissue. They're not carried along by the hemoglobin to
Mhm.
the lungs.
Mhm.
But at the end, I realized that the proton are binding again to the bicarbonate ions in the plasma. So
Mhm.
does that mean that the carbon gotten from the tissue is not being transferred to the lungs?
Okay, so the bicarbonate ions are moving freely in plasma from the tissues to the lungs, whilst the hydrogen ions are bound to hemoglobin. So they are carried on the hemoglobin to the lungs. Does that answer your question?
Yes, sir.
All right. Any other?
Yes, I have a question, sir. Hello, sir.
Yes, we are listening.
Oh, do you want to mean like
I
80%
can't
of
hear
the
you
carbon
well.
dioxide?
Bright.
Okay. Yeah, I said, do you want to mean that the rest of the percentage of the carbon dioxide, which is the 85%, are being exhaled?
Mhm.
Hello, sir.
I have not gotten you.
I said,
Mhm.
do you want to mean that the rest of the percentage of the carbon dioxide have been exhaled, since 15% have been taken to the peripheral tissues?
Mhm. So what we are saying is that only 15% of the carbon dioxide binds directly to hemoglobin. So this carbon dioxide is carried on hemoglobin directly to the lungs. And the rest of the 85% goes through this process of binding with water to form the carbonic acid and then broken down into the bicarbonate ion and protons. And the protons are what are carried on the hemoglobin, whilst the bicarbonate ion flows freely in plasma to the lungs. Understood?
Yes, please.
All right.
Sir, please, I have a question.
Okay. Emma?
And sir, please, what's the use of the protons and then the CO2 on the hemoglobin? Like, what's their function? What do they do?
All right. So it's not that there's any function that they are having on hemoglobin. They are being carried. So this hemoglobin molecule is carrying the proton. Of course, this proton, if these protons should be free in blood, in plasma, it will mean increase in acidity of the blood. So they are bound on hemoglobin, and hemoglobin is like a carrier of these protons into the lungs. But the bicarbonate ions, they can freely flow in plasma. So it's a form of transport of this carbon dioxide back to the lungs to be exhaled. Not that they are having any function on hemoglobin. Does that explain give you an understanding?
Yes, sir.
All right. Okay. So we continue, as I mentioned earlier, that we have what is referred to that as the allosteric effectors. So these interact with the hemoglobin molecule to facilitate the release of oxygen from the oxyhemoglobin in the tissues. So we have 1, 2, 3, 4 of them. The first is carbon dioxide, the protons, and chloride ions. Of course, we have seen the carbon dioxide and the proton already in action. And simply what that means is that in the tissues where you are having high amounts of carbon dioxide, that increase in the level of carbon dioxide in the tissues reduces the affinity of the hemoglobin to oxygen, so that the oxygen will be released to the tissues, whilst it will bind to the carbon dioxide to carry away. So in increased amounts of carbon dioxide, the affinity of hemoglobin for oxygen reduces. So it can release the oxygen and pick the carbon dioxide away. Similar way, we saw that in the tissues too, we are having the production of protons or hydrogen ions, and that means increase in pH, or is it decrease? Decrease in pH, because you are having pH less than 7 down. So the system is becoming more acidic. So the decrease of these protons, the increase in concentration of these protons, which is to say the decrease in pH, will also cause hemoglobin to have a decrease in affinity for oxygen. And that will mean it will have to quickly release oxygen so that it can bind to these hydrogen protons that are produced in the tissues to carry away. Is that clear for the two, carbon dioxide and protons, before we move on to chloride?
No,
Then,
sir. Yes, please.
okay. So now, chloride. So you see that once you are having in the cells more hydrogen ions being produced, which is increase in the concentration of hydrogen ions, and thus making the cell become more acidic, there has to be a neutralization of that acidity. And so chloride ions from the peripheral fluids then flow into the cell to neutralize those hydrogen ions, so that in the process of this increase in hydrogen ions in the cell, you have chloride ions moving in to cause that neutralization there. That is the function of fluoride ions in this aspect of respiration. So is the function of fluoride also understood? Is it understood?
Sir, please come again.
Okay, so what I'm saying is that in the cell, what I'm saying is that in the cell, where you are having this carbonic acid breaking down into bicarbonate and hydrogen ions, which means you are having now increase in concentration of hydrogen ions. And so we are talking about pH moving to the acidic region. Because you are having increase in acidity in the cell, there has to be a mechanism of neutralization of acidity in the cell. And the mechanism for that neutralization is that you have chloride ions that are in the peripheral tissues or the peripheral fluids that move into the cell to now bind with these hydrogen ions. So that if you are having carbon dioxide, being produced, and 85% of it is giving you these hydrogen ions, and that rate of which hemoglobin is able to carry this proton away from the cell is not that much that it can quickly carry all of it away. There has to be some temporal neutralization of this process. So you have chloride ions moving into the cell to neutralize these hydrogen ions. So in the increase of carbon dioxide in the cell, you have chloride ions moving more into the cell to neutralize the pH in the cell. That is the function of chloride ions in this aspect of respiration. Is that understood?
Yes, please.
Yes, sir.
Okay.
Yeah.
So we go further to look at, I think that we'll come to the last one, which is the 2,3 biphosphoglycerate. We'll come to it on this slide, the effect of the 2,3 biphosphate glycerate on the affinity of hemoglobin. We'll come to it over here. So let's keep that and we'll come to it here.
Hello, sir.
Yes.
Sir, yes, sir, please. This slide. Sir, please. I was asking, is that because of the presence of the hydrogen ion, the chloride ion will have to go and then neutralize.
Yes.
But we know that the hydrogen ion is a positive and the chloride is negative. So that means if the chloride goes to bind to the hydrogen, that will mean the formation of HCl,
Yes.
which is also acidic. So how is the neutralization done?
Okay, so it is actually not the acid, the molecular form of the acid HCl, which is acidic. It is the hydrogen ions, the dissociated form, that causes acidity. So if you have hydrochloric acid being dissociated in aqueous solution where you now have hydrogen ions freely, that is what causes the decrease in pH, the acidity. Understood? So that when you have chloride ions moving in to neutralize those hydrogen ions in molecular form, that will not make it, it will make it lesser acidic. Understood?
Okay, sir.
An acid will not be acidic if it is not releasing protons, if it is in its molecular form. Understood?
Yes, please.
So that's why you measure pH by concentration of the protons or concentration of hydrogen ions. All right, so let's move on. So all of these that we were talking about results into what is referred to as the Bohr effect. So the Bohr effect simply is that when you have pH being reduced to the acidic form, so like in the tissues. So if you have in the tissues where you have high concentration of hydrogen ions, and that will mean that the environment is more acidic, which is represented by the blue curve. So the blue curve, meaning that this is more acidic. So that is occurring in the tissues. So if this is occurring in the tissues where you have the acidity of the tissues increasing, you will have the saturation of hemoglobin shifting to the right, meaning that it will take a longer time for hemoglobin to get saturated with oxygen. In other words, we are only saying what we have said earlier, that the binding of oxygen to hemoglobin will be decreased, or the affinity of hemoglobin to oxygen will decrease in the tissues when you have more hydrogen ions present, because it will have to release the oxygen faster to carry the hydrogen ions away. So what it means is that in the lungs, so if you take the red line to represent in the lungs, so in the lungs, it will take you a shorter time, or the hemoglobin molecule has a higher affinity for oxygen. And so you see that the hemoglobin molecules get saturated earlier. But in the tissues, hemoglobin molecules will get saturated with oxygen later. In fact, what it means is that in the tissues, preferably, hemoglobin will release the oxygen and pick the carbon dioxide, or it will pick the protons out. So that same graph, that saturation graph, which we were looking at for the affinity of hemoglobin for oxygen, will shift to the right in tissues. Is it clear?
Sir, please go over.
Okay, so here is the thing.
Sir, please go over.
Yes, so here is the thing. We have the red line is representing the process of saturation, or call it the dissociation curve of oxygen in the lungs. Then we have the blue line representing what is happening in the tissues. Now, like we saw earlier, on the vertical axis, we see saturation of oxygen. So this is saturation of hemoglobin molecules with oxygen. And on the horizontal axis, we have what? The partial pressure of oxygen. Call it concentration of oxygen. Now, we see that the red line, which is representing what is happening in the lungs, where you have higher amounts of oxygen, we see that the hemoglobin molecules get saturated faster because the affinity for oxygen, the affinity of hemoglobin for oxygen is higher in the lungs. They are to bind to the oxygen, release carbon dioxide, and bind to the oxygen. And here you have lesser amounts of hydrogen ions. Now, when you come to the tissues, where you have higher amounts of protons, which means we are moving into a much more acidic pH. Here, we have the affinity for oxygen being lesser. So if the affinity of hemoglobin for oxygen reduces in the tissues, because the hemoglobin will have to release oxygen and pick carbon dioxide or protons, then of course, it will take a longer time. It will take a longer effort for the hemoglobin molecules to get saturated by oxygen. In other words, hemoglobin will prefer to bind with the protons, release the oxygen, and bind with the protons. And so you have this graph shifting to the right in tissues. Understood? Jose.
Yeah, sir. If I'm right, we know that in the tissue, oxygen is supposed to be released.
Yes.
Then why do we have that? Do we need for the affinity of oxygen to the hemoglobin in the tissue? Since they are being released, then why do we still have it? Because we know that in the tissue only 15% that is being bound to the hemoglobin, and others are transferred to the plasma. But I realize in the case in the curve, it's even more than 85%. So why is it so?
Okay, so now what we are looking at is that, yes, carbon dioxide should be released to the hemoglobin, and hemoglobin should be releasing the oxygen. It isn't that there is no affinity at all. It's not like there is zero affinity of hemoglobin to oxygen at any time. There is no such thing as, say, zero affinity to oxygen, but there is decrease in affinity and increase in affinity. So what we are looking at is that, since we were talking about an oxygen dissociation curve, you remember that earlier we saw this same curve showing that the affinity for oxygen in the lungs compared to that of myoglobin is such that, yes, at a certain point, you have myoglobin getting faster saturated. Now, we are now comparing that same affinity for oxygen in the lungs and in the tissues, but this time looking at only hemoglobin, not myoglobin, so that we are only representing the affinity for oxygen in these two scenarios, these two positions, in the lungs and in the tissues. Like you rightly said, in the tissues, the hemoglobin should be having less affinity for oxygen because it must release the oxygen and take carbon dioxide. And that is the reason why the graph shifts to the right. If you take the saturation of the hemoglobin molecules against the partial pressure or the concentration of oxygen in the tissues, you realize that the graph shifts to the right compared to what happens in the lungs. And that proves the fact that, yes, in the increase of protons, when you are having the acidity in the cell moving into the acidic region, where you have protons increasing in the cell, you have the affinity for oxygen decreasing. And if the affinity for oxygen decreases, it translates to the fact that it takes a more saturation of oxygen or a more concentration of oxygen in the cell to cause saturation of hemoglobin. So you see, so if you are taking the points one after the other, so you realize that here in the lungs, when you reach about, say, even less than 25 millimeter mercury, let's say that is the concentration of oxygen in the lungs. By the time you reach this 25, then hemoglobin is getting saturated. All of it, all the hemoglobin monomers get bound to oxygen. But when you come to the tissues, you realize that you have to have twice that concentration. So somewhere around 50 millimeter mercury before saturation of the hemoglobin can happen in the tissues. So it's like you need to have like twice the concentration of oxygen which you have in the lungs, in the tissues, to cause hemoglobin to actually get saturated. And that is impossible because oxygen is more in the lungs than it is in the tissues. So you put the hemoglobin in a position where it prefers to bind with carbon dioxide and to release the oxygen. Understood?
Yes, sir.
All right. So now we come back to the effect of 2,3-biphosphoglycerate on oxygen affinity of hemoglobin. Now, what this compound does, I think. Is somebody speaking? Okay. All right. So what this molecule does, so in fact, you find this molecule 2,3-biphosphoglycerate increasing in erythrocytes in anemic conditions. So now you see this same graph which we were looking at. The first, which is the red, that is for the normal hemoglobin, which is not bound to any 2,3-biphosphoglycerate. Then we have here some normal blood, which is bound to 2,3-biphosphoglycerate. Then the green is representing the blood of anemic patients with this 2,3-biphosphoglycerate. Now, what does this 2,3-biphosphoglycerate do? This 2,3-biphosphoglycerate binds with hemoglobin, as you can see in this structure. It binds to hemoglobin by some ionic bonding to the beta chains. And when it binds to these beta chains, of course, just like when you have oxygen binding to this hemoglobin molecule, it causes conformational changes so that you have the relaxed form. And the relaxed form means that what? It is oxy hemoglobin, meaning that it is bound to oxygen. Now, in this case, when you have 2,3-biphosphoglycerate binding to hemoglobin and making it look as if the molecule is oxy, but in actual fact, it is deoxy. So in other words, the system is being deceived. So what happens is that, let's say people in high altitudes will have high amounts of these 2,3-biphosphoglycerates binding to these hemoglobin molecules because there is less amount of oxygen present. And you don't want that when you have just the limited amount of oxygen present binding to the hemoglobin, you want the rest of the hemoglobin to be binding to carbon dioxide. You don't want that. So the rest of that hemoglobin, which is free, which is not having any any oxygen to bind to, binds to these 2,3-biphosphoglycerate to prevent it from binding to carbon dioxide. So that is like an adaptation to lesser amount of oxygen that is present. You find, so you find this happening in anemic patients. So you see that, yes, if you are having, of course, an anemic patient already, you are going to have some lesser blood counts. You are having issues with, the structure of your hemoglobin and things like that. And you are having part of that blood binding to 2,3-biphosphoglycerate, which is already in a relaxed form. What it means is that it will take you even more concentration of oxygen to cause the hemoglobin to get saturated. Since you already have 2,3-biphosphoglycerate binding to some of the hemoglobin present. Yes, in a normal blood with a 2,3-biphosphoglycerate at its normal levels, you find that you are having this graph in its norm, it will get saturated at a normal time. But where you don't have any 2,3-biphosphogylacerate bound to the hemoglobin, you even get hemoglobin getting saturated faster. So when you do all this comparison, you see the graph shifting more to the right with concentration of 2,3-biphosphogylacerate. When you have 2,3-biphosphogylacerate increasing in concentration, you find the graph shifting to the right. So the graph shifts to the right with increasing concentration of 2,3-biphosphogylacerate. Now, the binding of 2,3-biphosphogylacerate to the fetal hemoglobin is very weak. Of course, that is also understandable because if you are having these 2,3-biphosphogylacerate to have high affinity for fetal hemoglobin, it can cause suffocation of a fetus because if that hemoglobin binds tighter to this fetal hemoglobin, it will mean that it will starve the fetus of oxygen. So it is less for babies, for fetus. But in adults, it is some form of adaptation. Of course, in anemic conditions, as you see it here, you see that, yes, it means that there will be more of this compound present, more than normal, so that the test for these 2,3-biphosphogylacerate, if the concentration of it in blood is higher than normal, it will indicate some form of anemic condition. So it's a diagnostic It's a diagnostic method. Those of you working in clinical settings, if you watch some of the tests, the blood tests that you do, you test for this, the 2,3 biphosphoglycerate. Maybe they write it like this, BPG. It is the biphosphoglycerate that you are testing for, so that if it is high in concentration in the blood, it indicates some form of anemic condition in the blood. Any questions? All right, so it appears no questions. Okay, so let's continue. Now, we are looking at hemoglobin derivatives, which can be caused by some drugs. So some drugs, some free radicals, hydrogen peroxide in the body can cause the oxidation of the ion. Remember, the ion in hemoglobin is in its bivalent form. It's carrying the charge 2+. Now, some drugs, some free radicals in the body can cause the oxidation of this ion to the trivalent ion. And that will result in what is referred to as the methemoglobin. And the methemoglobin, of course, is having lesser affinity for oxygen. This is also an enzymatic reaction which is catalyzed by an oxidoreductase. This is methemoglobin reductase that can cause that. So some drugs can actually cause some hemoglobin derivatives like the methemoglobin. Now also, we have carbon monoxide. So carbon monoxide that we can get in some industrial settings. You know, in Ghana we burn a lot of things indiscriminately. We release a lot of carbon monoxide into the atmosphere, or you know, the engines of machines and automobiles releasing carbon monoxide into the atmosphere. Now it's interesting to note that hemoglobin has about 200 times more affinity to carbon monoxide than oxygen, so that in high amounts of carbon monoxide in the atmosphere, hemoglobin will bind tighter to carbon monoxide than to oxygen. And that is going to cause the hemoglobin derivative carboxyhemoglobin. And this, of course, will cause things like headache, nausea, vomiting, breathlessness, irritation. And this is understandable because if you have hemoglobin binding to carbon monoxide so tightly than it is to oxygen, it means that a lesser amount of oxygen is being carried to the cells, and especially to the brain cells. Is going to cause you a lot of these symptoms like the headache, your head will be turning, nausea, your face, you know, different breathlessness, of course, that's some kind of suffocation. And of course, in very high amounts, if this persists for a long time, it can result even into fatality. Remember, we are saying that carbon monoxide is bound about 200 times more than oxygen, which means that it will take a longer time or more effort for the hemoglobin to bind to oxygen. Of course, you have to be put on oxygen so that you increase the concentration of oxygen to actually bind with the hemoglobin for the carbon monoxide to be released from these hemoglobin molecules. Understood?
No, sir.
It's not understood.
Yes, please go over, please.
Okay, so what I was saying was that excuse me. So what I was saying was that you have some free radicals, some drugs, which can cause the oxidation of iron, the bivalent ion, which we find in the heme of hemoglobin, oxidation of it into the trivalent Fe3+. And this has lesser affinity for oxygen. And of course, this is an enzymatic reaction catalyzed by the methemoglobin reductase. Then the second one we are looking at as a hemoglobin derivative is a carboxyhemoglobin. So that in an environment where you have increased amount of carbon monoxide, it is interesting to know that carbon monoxide binds with hemoglobin about 200 times more than oxygen. So that if you have high concentrations of carbon monoxide and this bind to hemoglobin, which results into lesser amount of oxygen being carried to the cells, to the tissues, and especially to the brain cells, it will cause things like headache, like vomiting, some nausea. It will cause some form of heaviness in the chest or breathlessness, and of course, some irritation. The only way to deal with it is to put the patient or whoever is intoxicated with this carbon monoxide on oxygen. And so once you increase the concentration of oxygen, you are sure that oxygen goes in to displace the carbon monoxide in these hemoglobin molecules, and carbon monoxide will be released over time, whilst oxygen then now binds with hemoglobin to be transported to the tissues. All right. So we cast, before we go on with these hemoglobinopathies, we cast our mind back to what we did earlier on the genes. We looked at gene expression, which is divided into two, the gene transcription and then translation. So transcription giving us the messenger RNA, which is carrying the code from the nucleus to the cytoplasm, and then that's code on the messenger RNA is then translated into proteins. So what we have here, so the top, so the top here is referring to the genes that are responsible for the alpha globins. Remember, we said the hemoglobin is made up of the alpha globins. These are proteins that are bound to the prosthetic part, which is the heme. So if the top here, which means that those proteins, those alpha globins are coded for on a gene. So in a DNA strand, you have some sequences of nucleotides which will be responsible for the synthesis of these alpha chains. So we have here these genes coding for alpha chains. So we have the alpha 1, the alpha 2, and then of course this is epsilon, and this, this is zeta. Of this you'll find these in the fetus. You don't find this in adults. In very less amounts, you'll be finding the zeta. Okay, so what it means is that, yes, from these genes, you now have the alpha globins produced. The messenger. No, okay, here we have this diagram skips the process of transcription. So quickly it has moved to the globin chains, which is the protein, the alpha proteins. Then we have here, the down here, which is the gene responsible for the synthesis of the beta chains. So these beta chains, here too we have the delta, the gamma, and then the epsilon. So yes, the beta is what we find mostly in adults. So we have the hemoglobin A1, which you looked at earlier as having the 2 alpha and the 2 beta, which are the major ones. The rest, the delta, the gamma, the epsilon, these ones are not as common as as the alpha beta. So yes, here too we have the beta globins. These are the proteins. So the hemoglobins, which we looked at earlier, these are what the alpha 2, beta 2. This is the one we looked at as 90% hemoglobin A1 in adults. Then we have the alpha 2, delta 2, which we said, I think is less than 5% or so. Alpha 2, gamma 2 also is less than 2% or so. Then we have the zeta 2, epsilon 2. These two you find also in the fetus, and in some rare situations, you'll find these types of hemoglobin. So as we cast our mind back to that process of transcription and translation into the proteins, and then connecting it to the fact that these globin chains are proteins. And so it means that they are going to be coded for in a DNA. All right. So if that is understood, the first immunoglobinopathy, common one, which is mostly referred to, is the sickle cell. And that sickle cell on a molecular level is caused by just the missense mutation on the gene, such that for the position, say if you are counting the gene 1, 2, 3, you remember these are trinucleotides. So 3 nucleotides are coding for 1 protein. So the 3 nucleotides are coding for 1 amino acid. So on the 6th position, so if you are looking at this beta, the gene here, which is responsible for the beta globin, on the 6th position, where you should have glutamate or glutamic acid, you have that code being changed to rather produce valine. And that causes this crescent form of the sickle cell. Now let's look at it, in broken down more for us to see. Now, this is the hemoglobin A, the normal hemoglobin that we have. This is in the DNA, where you have cytosine, thymine, cytosine. And that will mean that on the messenger RNA, which is transcribed from the DNA, you have what? Guanine, adenine, guanine as the complementary codes, which are responsible. So these on the messenger RNA, guanine, adenine, guanine, on the messenger RNA is responsible for the amino acid glutamate. And this is the normal situation that you find in hemoglobin A. Now, the sickle cell hemoglobin, in the DNA, you find that just one of these nucleotides, the thymine here, is replaced by adenine. So instead of this pyrimidine thymine, you now have a purine adenine. And that translates to these on the messenger RNA. Remember, this is an RNA, and so you have this nucleotide being uracil. And this change alone in that code on the DNA now results into the synthesis of the amino acid valine on the 6th position on the beta chain. And that causes the sickle cell hemoglobin. Any questions? Traker, you want to say something? Charles Agboja, you want to say something?
Yes, sir. Please, I want you to go over this particular slide again.
Okay, the molecular basis for the sickle cell. So
Yes, sir.
I was saying, if we cast our mind back to gene expression, that is transcription and translation. So on a DNA strand, you have certain sequence of nucleotides, and out of it, you have the messenger RNA being produced. And from the messenger RNA, you have the synthesis of proteins. So every trinucleotide coding for one amino acid. Now, in a normal hemoglobin, the hemoglobin A, you have on the 6th position of the gene that is responsible for the synthesis of the beta chain, the beta globin. You have the code here being cytosine, thymine, cytosine. And that means the messenger RNA that is produced from that DNA strand will be guanine, adenine, guanine. These are the complementary bases to the bases that are on the DNA. Now, this code on the messenger RNA is responsible for the amino acid glutamate. And this is at the 6th position. So this is the beta chain, the 6th position. What is there? So if it is the beta globin, the beta protein, we are showing only the 6th position where you have glutamate here. Now, in a sickle cell, this pyrimidine, thymine, is replaced. So this is like some missense mutation, and so you have here adenine in place of thymine. And because of that, on the messenger RNA, instead of adenine, we now have uracil being the complementary base for adenine on the DNA. Now, on this messenger RNA, where you have this code, gene U gene, this is responsible for the amino acid valine. Valine instead of glutamic acid or glutamate in the normal hemoglobin. And this change alone of this amino acid in that globin chain on the 6th position will give you this condition of the sickle cell, the crescent structure of the red blood cell instead of the normal biconcave shape of the erythrocytes. Understood?
Yes, sir. Thank you.
All right. So we move on. So as we have understood that we have some other forms of these abnormalities. So we have the hemoglobin C, which will cause some mild anemia, again at the 6th position. That same glutamic acid in the normal hemoglobin will now be replaced with lysine. So remember, in sickle cell, it was replaced with valine, but here it is lysine. And if it's lysine, you have some mild anemia. So some sickle cell will be having these two. They can have the valine, and then some of them will be giving you the lysine, so that the person is sickle cell and at the same time having some mild anemia. That is the hemoglobin C. Here, then we have the hemoglobin D also being the mild, also giving some mild anemia. Here it is at the position on the gene 121st position, which is also supposed to be glutamate, but this time around it will be glutamine. Instead of glutamic acid, it is glutamine. That will give you the hemoglobin D. And then hemoglobin E, which is asymptomatic, on the 26th position. Here we have the glutamic acid also being lysine. So you see the difference between hemoglobin E and C is that here on the hemoglobin C, the 6th position, where glutamic acid is being replaced with lysine. But in hemoglobin E, it is at the 26th position that you have glutamic acid being replaced with lysine. So these are some other forms of abnormal hemoglobin forms. All right. Now, so looking at all of these things, there can be some forms of immunoglobinopathies due to some missing genes in a globin chain, or it can be that the gene is present, but the gene is non-functional for whatever reason. So like the gene is there, it's just silent and it's not functioning. Perhaps there are some other factors which are not present that will make that gene functional. So you have these different forms on the alpha globins, referred to as the alpha thalassemias. So for a normal person, yes, you see that these alpha genes so this is happening on chromosome 16. So chromosome 16 is having these normal genes, all of them functional. There is no clinical symptom. For some that are some silent carriers, you have just maybe one gene that is missing. That will give you no symptom. But you begin to have some minor anemia when you are having, say, 2 genes that are missing. On the same allele, you have 2 genes that are missing. You have some minor anemia. Some mild anemia will also occur when you have, these are 2 genes that are missing. There is 1 gene which is present, all right, but it is not functional. So already here, things are getting more serious. You have 2 missing, you have 1 which is not missing, but it is non-functional. Then you start to have some mild to moderate anemia. You can have normal life anyway. Then you can have things like you have 2 missing genes. Then you have 2 other genes that are present, but they are non-functional. And these can result into fetal death. And usually this will result as a stillbirth. So at birth, that's the baby is being born, the baby dies because of these alpha thalassemias. Any questions? Okay, so then we can move on to the beta. So similar things will occur with the beta thalassemias. So here you have on the beta globins for a normal person, yes, all the beta globins, the genes are functional, they are present, they are functional. Then we have some minor beta thalassemia where you have 1 of the beta genes that are missing, which means you are left with just 1 beta gene, which will cause some minor thalassemia. Then you have a major thalassemia when you have 2 of the genes missing for the beta globins, the synthesis of the beta globins. Okay, so of course this will result in some severe anemia, and in many cases a child born with these beta thalassemias might live for just about a maximum of 2 years. Okay, so that's it for the alpha and beta thalassemias. Any questions? All right, we have 9 minutes more, so we'll wrap up quick and then. So that's it for the hemoglobin and hemoglobinopathies based on these globin chains. The next time we meet, we'll be talking about hemoglobinopathies pertaining to the prosthetic group, especially.
Hello, sir.
Yes, Justice.
Hello, sir.
Yes, Justice, I can hear you.
This is Emmanuel Dake.
Okay, Iman.
Please, the beta thalassemia and the alpha thalassemia, how can you determine it?
How can it be? Iman,
How can it be
again, say how
determined
can it be? What?
clinically? How can it be determined
Okay, so
that you can prevent it?
Okay, so prevention is not something that we can think of because this is genetic. Remember, this is happening in the genes. So it's something that is inherited. So it's not a thing to be prevented. You only know about it and you manage it.
Hello, sir.
Yes.
Hello, sir.
Is it Justice?
Hello, sir. No, sir, it's Marvelous, sir.
Marvelous, I can hear you.
Sir, please, is there any control to the B thalamus? So is there any control to it?
Is there any control to what?
Hello, sir.
I'm listening. Is there any control to what?
To the beta thalassemia. So is there any control to prevent the child from dying within 1 to 2 years?
Well, if you have a fetus that is carrying is a carrier having just one gene that is missing, of course that will just be something minor asymptomatic. But if you have two genes that are missing, so that's what I was saying, that if you are having two beta globin genes missing, and this is, remember, this is genetic. And if this genetic condition which is being inherited, it's not something that really you can say taking some drugs and things like that, and then you'll be okay. So many of these, yes, you will know about it, but you will not have any power to do anything about it. You just know that it is there, but what can you do? It's genetic, so there's nothing much you'll be able to do about it. So far, there hasn't been any form of gene engineering or anything of the sort to be causing any changes in these things. I saw
Hello,
some
sir.
other hand up.
Hello, sir.
Sir, it's Justice.
Yeah, Justice.
Hello, sir.
So many people are
Yes,
talking at
sir.
the same time. I will mute all of you, and then Justice will unmute. Okay, so Justice, now you can unmute and talk.
So I raised my hand.
So you can talk. We have 5 minutes more. Justice.
Sir, please, is methylhemoglobinemia a hemoglobinopathy?
I don't even know what that is.
Because you didn't mention it.
I don't know what it is.
Sir, the ion existing.
There is what?
Sir, like the ion existing in a 3 platform instead of a 2 platform.
That's methemoglobin. I mentioned it.
Yes.
Do
we have a defect for it? Do we
Does it
have a
have
defect for it? A defect.
a defect?
Yes.
Meaning, what do you mean by defect?
So, you said the ion is supposed to be in a ion 2 platform. So
Yes.
if it exists in the iron 3 plus form. Is it a defect or it's a normal?
It's a defect. So I mentioned that it reduces the affinity for oxygen.
Okay.
That's you are talking about the hemoglobin derivatives. When you have that oxidation happening in the ion of the heme, you have a reduction for affinity for oxygen.
So can that also be considered as a hemoglobinopathy?
Yes, it's a hemoglobin. No, that is not a hemoglobinopathy, but it's a hemoglobin derivative which is forming as how do you call it? Due to some oxidative reactions in the body that will be causing that. Well, yeah, you may classify it as a structural, some structural difference. So somehow, but it really does not result directly into any form of a disease condition. So we shouldn't be classifying it as a hemoglobinopathy. We can only talk of it as a hemoglobin derivative, which can occur when you have some oxidative reactions happening in the body. Because usually when we talk about the hemoglobinopathies, we will be very much referring to either the differences, these genetic differences which may occur either to the alpha or beta chains, or you have some really permanent changes to the heme structure itself. Yes. Who was speaking? Was it Justice? Okay, so I've seen
Yes, sir.
the hand of Alex.
Okay, so my question is, with the thalassemias, can stem cell transplants be of help?
Stem cell transplant. Well, maybe. I don't know. Have you read anything on it? It will mean you have to be doing the transplant over a period of time, to be replacing the normal cells that are already present. You have to do this over a certain period of time to replace those stem cells for the production of the normal hemoglobin.
Yeah.
That will be an expensive procedure.
Okay.
Have you seen anywhere that there is some stem cell transplant which is used for the remedy of such?
I watched a movie and I saw something like that inside.
So there are some stem cell transplants. Okay. Well, there is a possibility. Some of these, sometimes they are being thought of. They may look fictional, but it can become successful at a point.
Okay,
but I haven't heard of any. I don't know.
Okay.
Charles? Charles, so you are the last person. It's exactly 11 o'clock. Okay. So Charles has muted. So that means Charles doesn't have any question. And it's exactly 11 o'clock. So we'll put our meeting to a close now till we meet the next time. Okay. So Moses and the others, you can keep your questions when we meet the next time. You can ask. We have been online for 2 hours. And
okay, sir.
We can understand that many will be tired. So the next time we meet, okay?
Okay, sir. Thank you very much.
All right. So all the best. Bye.
Thank you, sir.
Bye, sir.